Hey, so this is the section from chapter 1 that's going to be an introduction to some of the T cell stuff that we're going to talk about. So we'll go through this, it won't take but a minute. All right, you remember these guys, right? Yeah. MHC class 1, MHC class 2. The big difference in the drawing is that the one has two legs, the other has one. MHC class 1, one leg, two legs. And they aren't legs, they're transmembrane helices, but you know what? They look a bit like legs. They can each present peptides, and remember, class 2 can present bigger peptides than class 1. They tend to average around something like 14 amino acids, but it's actually unconstrained. They can hold on to much bigger things occasionally. So, all right. Right. And again, review. Let's say you got a cell. Let's say the cell gets infected. The point of the immune system is to find that cell and get rid of that cell, because once a cell is basically working for the virus, growing more copies of the virus, you need to stop that as fast as possible. So that is the idea. And yeah, you remember how it happens. So the virus comes in, that's great. The virus will start to copy itself. The viral copies will have RNA. That RNA will make protein, and the protein is going to make new viruses eventually. Some of that protein, though, gets chopped up because the proteasome is always watching and always chopping things. And when the proteasome chops up a little bit of virus, it may fit into an MHC class 1. You can see the one leg. And if it does, then it can present that peptide at the surface. And this is just the cell's way of showing a little bit of whatever it's doing at the moment. It's kind of like the idea behind open source, and that if you can see a little bit of what's going on inside, then things aren't scary, and secretive and bad. So, yeah, your body is basically a big open source principle biological machine. Okay. What's the goal here? Well, we want to get that infected cell, and so here's the cell surface with little viruses coming out. And let's see, those look like influenza virus to me, but I may be wrong there. But it would pile up at the cell surface like that. Let's see, it could also be HIV, I guess, but immature HIV. So immature. Let's see. The goal is for a cytotoxic T cell with this sort of killing effector function to come along and to either destroy that cell or cause the cell to destroy itself. And cytotoxic T cells can actually do both of these things. And we will get into this. This is all very much a part of the section that we're going to cover. So here's an example of virus-infected cell, and this is a T cell coming up, and it makes contact with it, sort of touches it with these little, like little fleshy fingers, pseudopodia, and on the ends of those are the various proteins that it needs to survey the cell. So it's going to have, if it's a cytotoxic T cell, it's going to have some T cell receptor, it's going to have some CD8, and it's going to have a variety of other attachment proteins to help it sort of stay on there while I can check out that cell, make sure it's not up to no good. And then it can either roll on and do something else, or kill that cell and, yeah, then go do something else. It's good to be the T cell, yeah. Okay, cytotoxic T cells recognize that MHC class 1. Like, yeah, we know this, right? Yeah, for sure. And then it has several ways, and there are a couple charts coming up with some of the ways that cytotoxic T cells can kill, and you'll actually find more in the book if you're interested in that. Granzyme is one of the things, but there's a whole great big list. Also, the T cells have molecules on the surface that interact with what are called death domain proteins. And these are like little secret buttons that you can press on the outside of a cell that will cause the cell to go into apoptosis. So, useful thing to have, because, yeah, when cells go bad, you need to shut them down before they become cancer or virus factories or what have you. All right. So that's the cytotoxic T cells. Then you got your Th1 cells, which are going to be our kind of mainstay, the thing we want to clear up an active infection, basically. And Th1 cells, in addition to their other functions, are mainly about activating macrophages. So in this case, they're showing a macrophage that had some mycobacterium inside of it, and when the helper T cell activates it, it will actually cause the lysosomes to fuse with these sort of other vacuoles inside. And they'll end up digesting some of the mycobacterium, and you will end up presenting peptides from that to other T cells, to other B cells, and in general, this allows, that component of the immune system to make an immune response. So Th1s, one of their big things is activating macrophages. Now, they'll also fire up NK cells, and they're going to release interferon gamma, which is going to shut down the Th2 pathway to some extent. But we'll get into all of that. There are wonderful charts coming up. And as a suggestion, I think for this T cell bit especially, there are a lot of pathways, and there are a lot of lists, like where it'll say, this is the cytokine that does this particular thing and comes from this particular cell. I think this is a real good one if you want to build, whatever the online flashcard site is, or if you just want to get some 3x5 cards and make yourself some flashcards. This would be a real good way, I think, to study this. Otherwise, you'll get tangled up. You'll remember the general outline, but some of the names will escape you. So that's a suggestion anyway. Yeah. All right. Let's see. Let's make that a little smaller. There we go. And make it a little moved over. There we go. Okay. We'll introduce an additional kind of T cells. So, Th1s, Th2s, Th17s, and, T regulatory cells. But there are also T follicular helper cells. And so these are going to hang out in the B cell follicle, where all the young, naive B cells gather together and, trade antigens, basically, or wait to see if they can find an antigen. It's like, I don't know, like immigration or something. I don't know. That's not a great analogy. But what they do is they are going to be mostly confined to these B cell follicles. So they're going to live in there. Otherwise, they are very similar to Th2 type T cells in that they are supporting B cells. They're releasing interleukin 4, for example, which is going to make more T helper cells. So the Th2s, they make slightly different cytokines, and the Th2s are able to run around in the bloodstream. So they're like a mobile version. The T follicular helpers are the sort of stay-at-home, staycation version of the helper T cell, Th2 type. Yeah. All right. So, for example, here's our picture, and now we're putting some context to it. And shown at the bottom is actual microscope pictures of this going on. So we've got cells here stained up in blue, and they've got little bacteria inside them. And this is what like the tuberculosis bacterium would look like inside of cells. And so those are the little pink things. And the idea is that when a macrophage is stimulated, it will actually dissolve whatever it is that it's just eaten, and it'll accidentally dissolve other things that are inside. It may end up chewing up some of its mitochondria. That's okay. It can make more. Yeah. And this is by merging lysosomes with other compartments inside the cell, and then processing a whole bunch of antigen that's in there, and then presenting it on the outside. And so if a Th1 cell has a T cell receptor that can recognize the antigen presented on MHC class 2, because macrophages are best at MHC class 2 presentation, so talking to helper T cells, then they will release interferon gamma. And interferon gamma is going to stimulate the macrophages, and it's going to have effects on a whole bunch of different cells. And we're going to activate interferon gamma pathways. These are going to have effects that include sending the cell into an antiviral state, so it's harder to infect and more resistant. Basically fights infections a little better, slows them down. and lots of signaling. It's all different kinds. So helper T cells, Th1s anyway, their job is to go out and find some macrophages. And because macrophages are just always eating stuff. The name literally means big eater, and they're big and they're eating stuff all the time. And when they're stimulated, they're going to dissolve everything they've eaten, and they can then prime a lot of immune responses. They can prime helper T cell responses, and those helper T cells can theoretically go and do a bunch of things. Yeah, talk to B cells, fire up some natural killer cells through cytokines. Indirectly activate CD8 cells. That's going to be one of the downstream effects of interferon gamma. All right, and that's the end. That's the end of this section. So there's not a lot of test-worthy content in here, but it's an introduction. That's all it is. So next we're going to jump to chapter 9, which is kind of the big picture, how stuff works, what these cells are trying to do. Then we're going to go back to chapter 7. Chapter 7 is all about the signaling cascades, and that's where the meat of the stuff on the test is going to come. And I want to make sure we have enough time to do a lot of that. So we'll go fast through 9, take our time on 7, and then we'll do a little bit of 11, and that will round out the course. So there you go. See you next video.